While that's being sorted out, our last trainee speaker is Agnieszka Burzynska. I may have said that wrong. She's at the Human Development and Family Studies and Molecular, Cellular, and Integrative Neurosciences, director of the Brain Lab at Colorado State University, and they'll be giving their talk on combining quantitative MR and symmetric fusion analysis to study the myelin hypothesis of cognitive decline.
So my name is Agnieszka Burzynska, and I will be co-presenting with my graduate mentee, Andrea Mendez Colmenares. So thank you so much for introduction, and we can start right away, and I'll try to catch up with time. So for decades right now, the dominating hypothesis about the pathophysiology of the Alzheimer's disease has been the accumulation of amyloid plaques and tau tangles. And as a result, the development of drugs was targeted to reduce the amyloid burden in the gray matter. However, as all of you may know, that to date, those treatments that are available or in development, they still have limited effect on clinical outcomes. Therefore, in my laboratory, we are interested in exploring alternative hypotheses of Alzheimer's disease. In this case, we are studying the myelin hypothesis. And this hypothesis posits that the amyloid and tau deposits may be a homeostatic response to an earlier attempt of myelin repair and failed axonal transport. What this implies is that white matter pathology may precede, at least to some extent, gray matter pathology. And if this is the case, we could consider myelin as a new future target for therapies, and also we could it could give us a new window for earlier, more effective interventions. So the known histological changes in both healthy aging and Alzheimer's disease include predominantly loss of myelin, the degeneration of the whole myelinated axons, as well as upregulation of microglia and astrocytes. However, as noted in earlier talk by Bogdan Dragański, the histological evidence for those changes is relatively scarce. This is because the evidence relies on the small number of specimens. Often the analysis was limited to just a few regions, and this is often related to the relatively poor fixation in the white matter. On the other hand, when we look at the in vivo studies, most of them have used DTI, and as we also know from previous talks, DTI, of course, the model has a lot of limitations. In the recent years, there's definitely more interest in using more advanced techniques, such as quantitative MRI. However, majority of studies used only a single technique, and moreover, very few of those new studies on aging or Alzheimer's disease correlated MRI indices with cognition. As a result, to date, there's still poor evidence for the myelin loss in vivo and also for its impact on cognitive decline. So the key problem is that there's no single MRI technique that can characterize white matter tissue in vivo, and we have heard a lot about this in previous talks, that there is still no one-to-one mapping between MRI parameters and underlying histology. So we are taking currently an experimental approach in which we are acquiring multimodal set of different techniques that are sensitive to water tissue interactions at multiple temporal and spatial scales. And so the techniques that we are acquiring include, for example, the novel qMT, the conventional myelin water fraction. We wanted to use another technique, but we were getting some artifacts on our scanner, and also the Chi separation susceptibility mapping. So novel techniques, and here I wanted to acknowledge John Collee and his colleagues that supplied us with those free, pretty much still in development techniques. And in addition, we are also acquiring DVI, with the aim to do no demodeling. However, after listening to Ilyana's talk, I'm definitely open to trying also other models. And then once this data is acquired, we plan to conduct symmetric fusion analysis that should give us more confidence in estimating the age differences. So the current studies on healthy aging, age differences in the microstructure. And the type of multimodal analysis we are using is the symmetric fusion. And this is the type of multivariate analysis that allows for using the highest amount of information across different modalities. And this allows us to capitalize on the strengths and help overcome limitations that are implicit in each method, especially knowing that each method really relies on a certain model that has its own limitations. And in particular, we are using the multiset canonical correlation analysis with joint independent component analysis developed by our collaborator Vince Calhoun. And this is because the mCCA maximizes the correlation between different image types, and joint ICA provides superior source separation. So in a very gross oversimplification, what are our expectations here is that the fusion analysis will yield independent components, and the analysis of their content and spatial localizations in the brain will give us some more information about underlying microstructural age differences. So, for example, looking at the first component, if we will see that there is a colocalized decrease in different measures of myelin content, each of which is not perfect, but if we see this colocalization, so it looks like there's the same source, we will have greater confidence to expect that this particular region is affected by demyelination. On the other hand, if we would see additional decrease in the intracellular volume, so intraaxonal volume fraction, we may expect that there's also loss of the entire myelinated axons. But there may be also modality unique components that, for example, will only show changes in orientational dispersion index that would signify only changes in orientation of the fibers or composition within a voxel without major changes in myelin or axonal integrity. And what is important that we will be relating each of those independent components to a comprehensive set of cognitive measures, because here I wanted to highlight the importance of the context, because even if in the future years, I hope so, we will improve the mapping between MRI parameters and underlying histology. However, the meaning of the microstructural changes is really dependent on the context, so the type of neurodegeneration, because demyelination or gliosis may have different effects on brain function or cognition in, for example, Parkinson's disease, Alzheimer's disease, or multiple sclerosis. So that's why when we study healthy aging or Alzheimer's disease, for us it's very important to understand the impact of particular microstructural changes on cognition. And here I'm going to hand over my microphone to Andrea, who will guide you through a short demonstration of the fusion technique to studying age differences in just DTI parameters, because our data that I just presented is still being actually collected and pre-processed.
Thank you so much. So here, I just want to show the first demonstration of this fusion approach using mCCA or multiset canonical correlation analysis and jICA or joint independent component analysis. So first, I want to show you how in this figure, how the fusion model works. So mCCA and jICA, what we want to obtain from that is to obtain covariate patterns of these DTI features, FA, MD, RD, and AD, and see how these covariate patterns distinguish between young and old, as we see in the picture on the left side. So to start, what the model does is that we have 4 different DTI features that we want to decompose into parts. So in the first step, we use mCCA or multiset canonical correlation analysis to break these features into a series of components based on their correlation and variation across the different modalities. So in the initial step, mCCA, what it does is that it projects the data into a space that links the modalities to maximize their covariation, to maximize the correlation between the different modalities. We then use joint ICA, which is on the right of the image, to project to obtain independent sources or independent components where each independent component represents a different mechanism to distinguish between young and old. After we have done the ICA, we look at the quality and the stability of these components so that we can ensure replicability. Then from the ICA, we extract ICA loadings or mixing coefficients for each modality. We can move to the next slide. Thank you. So this is the demonstration of one of the components with the highest stability or replicability index. So in the top, the spatial map shows a modality shared component between RD and AD that shows a covariant pattern of increased and decreased RD and AD that is very good at distinguishing between young versus old. So in this image in the top, the red voxels, for example, show white matter regions where mixing coefficients are higher in older versus young. For example, we observed that in RD showed an increase in older adults in the internal capsule, in the splenium of the corpus callosum, and the prefrontal white matter. Then what we did after we looked at the spatial maps is that we correlated those mixing coefficients or ICA loadings with cognition, again, to look at how this associates with important cognitive abilities. And we found that both RD and AD correlated very well with executive function or fluid abilities, as well with processing speed. What is important from this is that in the same sample, we looked at unimodal analysis, looking at the DTI features separately and their correlations with cognition after correcting for age, and this was no longer significant. So we can say then that this is the first demonstration of this fusion model, or mCCA+jICA, using white matter only, and it's promising in identifying joint or shared age difference across the different feature maps, and has superior association with cognition when compared to unimodal analysis. So this has potential for identifying new patterns of white matter aging with a data-driven approach. As well, we developed some quality measures that also ensure replicability and reproducibility of our results using the fusion model.
Thank you, Andrea. So our next steps will be to conduct similar multimodal fusion using the newly acquired qMRI data and correlate it with cognition and indices of health. Especially our lab specializes in using sensor-based physical activity. So we want to know how physical activity habits relate to health of white matter, also knowing that white matter is, the maintenance of white matter is metabolically expensive. So physical activity and metabolic health should make a difference. And we are also looking into the validation and extension of those current analysis to the canine model of Alzheimer's disease. And I wanted to thank the Alzheimer's Association, National Institutes of Aging for funding this experimental study, as well as John Khalif, Lee, and Vince Kaloun for providing technical support for the study. Thank you.
Great. Thanks so much for the interesting talk. Was there any questions, Olivier?
No questions yet in the chat.
I guess I can get started with potentially one. You sort of introduced that you have an accumulation of potentially proteins such as tau with aging or disease state, and that's combined with demyelination. So something like MT, those potentially could be canceling each other out. You have a loss of one macromolecular pool and an addition of another. So how are you planning to disentangle this with your acquisition protocol? Or are you planning to look at that?
Can you just repeat which exactly molecules you meant?
Well, just sort of protein accumulation that might happen with aging, such as tau or amyloid, which will increase your macromolecular pool. And then that's potentially combined with the demyelination, which so then, I mean, who knows where the balance will end up falling, but it potentially could even out.
Right. No, this is a great question. So I wanted to clarify that we are actually looking only in the white matter space. Of course, our data will allow us to link it later to the gray matter. But the whole idea is that typically, you know, histologically, also using PET, what is typically looked at is actually the tau and amyloid in the gray matter, but we want to look at the earlier processes. And actually currently we are looking into age differences in white matter. So we are not yet including a clinical sample. So first we are making this proof of principle, but we are really looking into the white matter, for example, to look whether, you know, what are the real processes of the ideally healthy aging. Actually, our sample is a bit overly healthy so that we do not have any other conditions. So we have very strict exclusion criteria. And the next step will be to include clinical samples. But again, we are really trying to look into possibly earlier step because, you know, there's a lot of it's a lot of also unknown where, for example, the tau signal from PET comes from, but there's some evidence, a lot of it comes already from the so-called ghost neurons, because the tau has to be released from the dying neurons to be captured, in many cases by PET. So we are really trying to look perhaps decades before even into very early changes in the white matter that may result much later into those typical Alzheimer's disease biomarkers in the gray matter that are currently kind of definitive and diagnostic.
Very interesting. Thank you.
Thanks. One more question in the chat. Malash Gravardi asks, can you provide a bit more rationale for the CCA plus ICA analysis? This will give you some neuroanatomical signatures, but how are they going to provide increased specificity to neurodegenerative processes?
It's a great question, and I don't say that we will really increase specificity because this would be too bold of a statement. What we rather hope for is that it's a bit like, you know, using ICA, it's a bit like you trying to get sources from multiple microphones. So in a way, our different techniques will be, you know, different sources of voice, but then we use the mCCA and joint ICA to separate kind of what type of major source they are representing or voice, right? So I would not really use specificity. I would rather say that we are more likely to just have slightly more certainty in detecting demyelination, for example, because, you know, in some techniques may still detect myelin, but it may be myelin debris. That's why we are, you know, using a combination of the myelin water, the macromolecular fraction, and also susceptibility. But it is a bit experimental because, as you know, Jung-Ho mentioned yesterday that, you know, the sensitivity to intra-individual differences in the chi separation is yet to be established. So we'll be also testing this. I don't know if I fully answered your question, whether your question was more specific to the mCCA joint ICA model per se. So I don't know.
I think he said it was good. So just in the interest of time, we'll